The Clostridium tetani neurotoxin is released from the bacterial cell as a 150 kilodalton inactive precursor protein. Extracellular proteases cleave the precursor polypeptide into a 50 kilodalton light chain and a 100 kilodalton heavy chain that remain linked by a disulfide bond. This structure is very similar to another clostridial neurotoxin, botulinum toxin. The amino-terminal light chain is a zinc-binding endopeptidase, while the heavy chain consists of two primary domains: the receptor binding domain at the carboxy terminal end and the translocation domain in the amino-terminal portion of the heavy chain. The receptor binding domain is further subdivided into two functional areas. The terminal carboxy end is responsible for binding to specific cell surface receptors. Adjacent to this area is a carbohydrate binding site that regulates intracellular movement of the toxins. Because both Clostridium tetani and Clostridium botulinum toxins bind to host cell surfaces with sialic acid containing receptors, toxin specificity is determined by binding to a second cell surface receptor. For tetanus toxin, that second receptor is a 15 kilodalton glycoprotein. The lipid and protein receptors concentrate in lipid-rich membrane areas, which facilitate transport of the specific toxins into the cell. Tetanus toxin enters the cell in non-acidified endosomes and is rapidly transported in a retrograde fashion to the motor neuron soma in the spinal cord. Once in the soma, acidification of the endosome lumen leads to a conformational change in the amino-terminal portion of the heavy chain, insertion of the heavy chain into the endosome wall, and translocation of the tetanus toxin light chain into the motor neuron cytoplasm. The light chains of tetanus and botulinum toxins inhibit neurotransmission by interfering with the release of neurotransmitters present in synaptic vesicles. The vesicles in the neuron carry neurotransmitters to the cell surface, where they fuse with the neuron plasma membrane and release their contents to the extracellular space. Fusion of the vesicles with the plasma membrane is mediated by interaction of proteins on the surface of the membranes. The proteins are members of the snare family of membrane proteins. Synaptobrevin on the synaptic vesicle interacts with SNAP25 and syntaxin on the plasma membrane to form a stable complex, leading to membrane fusion and release of the neurotransmitters. Both tetanus and botulinum toxins cleave synaptobrevin, preventing membrane fusion and release of the neurotransmitters. The effect of this inhibition is determined by the targeted synaptic vesicle. Tetanus toxin interferes with fusion of vesicles transporting inhibitory neurotransmitters such as glycine and gamma-aminobutyric acid, leading to unregulated stimulation of muscle activity and spastic paralysis.